pumps. Our cells are very greedy for potassium, and they don't like sodium. And then the extracellular space, all the space outside the cells, is really, really full of the opposite of that. Okay? And having those gradients of potassium and sodium generates a whole lot of electricity. Every single cell in your body has a voltage across its cell membrane. Okay? So our bodies are electric. And bioelectricity, this is what we call bioelectricity. This is the subject of the course that I teach. So you should be in my class. You should teach my class. Bioelectricity directs key functions in the body. Key functions like information flow in the nerves, and as I mentioned, the heartbeat. Okay? And so this is not a very new concept, actually. This has been known for about 100 years at least, since the first EKG technologies were developed. There's all kinds of practical applications for this, okay? Not just in stimulation. So, you know, there are deep brain stimulators on the market, cardiac pacemakers, bone healing devices, and some of these, you know, beauty treatments. But also measuring, okay? If you had if it's been suspected that you have a stroke, you will get what's called an EEG, measure the brain waves, okay? An ECG, an electrocardiogram, measures the waves in your heart. EMG, electromyogram, premature babies, doctors will measure the electrical currents associated with the breathing of these premature babies and decide, you know, how are they doing? Or, you know, lie detector tests. If this were my class, we'd actually get to see a little video of him failing very badly. So just to say, this is something that's, you know, a known concept. We know this. And so, you know, for my research, so I mentioned we build these enabling technologies. I'm actually showing you in much more depth than I would ever show a general talk, so ask questions if it doesn't make sense. But, you know, a lot goes into making these what we call enabling technologies. I keep calling them enabling technologies. Do you know why I keep saying that? Because the cells are actually doing everything. All we're trying to do is create a home for those cells, a happy home for those cells, so that the cells will just continue their work. Because the cells really do everything. The cells are the tissue engineer. So, you know, it sometimes feels like a bit of an identity crisis for us engineers, because all we're doing is building and enabling technology. The cells are kind of taking it off from there, right? But that aside, there's still plenty of engineering work to be done. I've had to program stimulator circuits. Stimulator circuits that provide the electrical signal that copies what the cells would feel in the body. I built these bioreactors. So I'm outfitting petri dishes with electrodes. I've had to do a lot of studies to study the electrochemical interface between the electrodes and the cell culture media, the cell's food, because that's going to impact the amount of charge that the cells feel. There's a lot of engineering work that does go into this, despite the fact that the cells are doing everything. Okay. And so this is this picture here on part C here. Well, part B is a picture of a petri dish that's outfitted with these electrodes. And if you zoom in really close, you can see a little piece of engineered tissue. Okay. And what's really so what happens? These tissues are about the size of a mini marshmallow, actually. And if you grow, these are two samples of engineered cardiac tissue that I engineered in the lab out of rat cells. Okay. And if you grow these cells just on a sponge, give them that schmutz that we talked about before. I don't know if you can see, but in the left-hand corner, there's like a little bit of nudge, that little blob up on the upper left-hand corner. There is some beating. These cells are beating a little bit, okay? And that's amazing, first of all, because this petri dish is really nothing like what these cells would experience in the body. But what's really amazing is that if we actually grow them in the petri dish that I showed you just in the last slide, and we grow it for 1 week, we grow this piece of tissue for 1 week with the electrical signal that's mimicking what the cells would feel in the body, it beats so much more. Isn't that amazing? I never get sick of it. I really do. So by mimicking what these cells would feel in the body, we can get so far. And so, what I spent a lot of years doing actually was asking myself, okay, what type of electrical signal would be best for these cells? Okay, so what kind of voltage, what kind of frequency? So I modulated those. I asked the question, what or how. That was a how question. How can I give these cells what they need? What do they need? So this is engineering. I modulated different voltages, different frequencies, and well, you know, got some answers. Actually, the answer was that these cells beat best with a stimulator duration of 2 milliseconds at 3 hertz at 3 volts per centimeter. And you're like, okay, what's the big deal? And what was really, really, really interesting to find out from that was that 2 milliseconds was the shortest duration that was necessary to get the cell to beat. 3 volts per centimeter was the minimum voltage that was necessary to get the cells to beat. And 3 hertz was really close to what those cells would have experienced in the body. These were rat cells. Okay? And so the lesson that we took from this is that if we give the cells what they need, they thrive. And not too much, not even a good thing. Okay? These cells just needed a little bit of a nudge, not a kick. Okay? And this was a real lesson for us. That cells not only do all the work, but they can do so much with so little. Okay? And these are the cells that live in your bodies. Okay? These are the cells that create life for you every day. I mean, this is just something that I feel can really, that we can, lessons that we can take with us as we live. You know, because we live in this body that these cells create for us. Anyhow. So, you know, we did a lot of other studies too. We studied embryonic bodies. These are embryonic cells. And we studied how they were developing, because electrical signals are actually really important in developing embryos. They've been implicated in patterning, where the left side and the right side of the body are, and all sorts of other things. And so we studied embryonic bodies with electrical stimulation as well, and saw that indeed, that, how do I show this? It's a little dark. But that these striations here of troponin is a really important cardiac protein. We saw that those striations, those patterns, were much better developed in electrically stimulated EVs, embryonic bodies, than ones that weren't. We did other experiments where we applied a different kind of electrical signal.
And is that all still being done kind of the lab level, or is it?
Oh, those were human cells. Yeah, and these are actually also human cells. These are human adipose-derived stem cells. Do you guys know what adipose tissue is? It's fat. Most people aren't really aware that we have tons of stem cells inside our fat tissue, healthy fat tissue, and they're really important cells. So fat is not all that. But if we electrically stimulate these fat cells, do you see how they move? You know, there's an electrical signal that's going from top to bottom here. The cells are aligning and elongating. By the way, this electrical signal is copying the same electric field strength that your body experiences whenever you have a wound. Okay? Stem cells are the cells in your body that repair. And so if we can copy the electrical signals that these cells are perhaps interpreting as a wound, then maybe we can get those cells to repair our bodies more effectively. Okay, so that's the rationale behind that. Oh, sorry. Having clicker problems. And you know, not only do they align and elongate, but they also have very different patterns. You see, these pictures, they're just so beautiful to me. I hope you enjoy them. But these little stars, these green stars, do you guys know what that, can you postulate what that might be? If I didn't tell you anything, would you think these were cells, by the way? No. They kind of look like planets. I don't know. In a future life, I would love to have a coffee table book of these kinds of images, by the way. But these are connexons. Those little stars, they're called connexons. They're the proteins that connect cells to neighboring cells. And it's like a little core, so that charged particles can move back and forth. And so they end up, when we stain them with these immunofluorescent proteins, they end up looking like stars. And they're just beautiful. But those patterns change with this electrical signal.
What magnification is this at?
This is at 20x. By the way, if any of you guys are interested in any of this in further detail, I'm really happy to share. This is all published work. I'd love to introduce you to this work in more detail.